And we come to the second talk, and I ask the audience to take seats. I think there are still some seats, especially here in the front.
Can you please take your hair?
So the second speech will be from Norway, and the speaker is Professor Sandvig, and she is going to talk about entry of nanoparticles into cells, mechanisms, and consequences. The floor is yours.
Can you give me the first wrong?
Yes.
Go back.
Can you put the other presentation? No, it's
No.
the wrong one. It's the next one.
It says Sandvig on the first slide, if you can find it. Oh, Sandvig, CLINAM. It got a strange number on. Hopefully that's my lecture.
That is this one. This one.
Could be that one. Yeah. Okay, thank you very much. So I'm going to talk about the entry of nanoparticles into cells, mechanisms and consequences. And as you know, many particles, at least with drugs, need to be taken into cells by endocytic mechanisms before they release the drug that acts on the cell. And nanoparticles use several mechanisms, and they can even induce their own uptake by mechanisms which are not normally present in the cell. So they are taken in by clathrin-dependent and clathrin-independent endocytosis, and it can be both dynamin-dependent and dynamin-independent processes. And you may know that there are now more than one type of macropinocytosis, one that is dynamin-independent and another one that's dynamin-dependent. And in most cases, what's taken up end up in early endosomes, and then it can be further transported into the cell. We still see articles where people try to direct their nanoparticles to caveosomes, which have been reported to be neutral, but these have turned out to be artifacts, so one should be aware of that. So, of course, many of us study now a non-polarized cell, but the picture is even more complicated in a polarized cell. You do have the same type of different mechanisms, but for instance, clathrin-independent uptake on the apical side can be regulated in a manner which is very different from what you see on the basolateral side. In a polarized MDSK cell, for instance, you have a large upregulation with protein kinase C on one side, not on the other side. And caveoli can be found on only one side, in this cell type on the basolateral side. So one should be careful with conclusions about caveoli uptake. So it can be difficult to study the interactions between different endocytic pathways because they are partly co-regulated. There is, for instance, a co-regulation of caveoli and CDC42-dependent fluid phase endocytosis, which can both be regulated by phosphocaveolin 1. For instance, if phosphocaveolin 1 goes up, then CDC42-dependent endocytosis goes down and fluid phase uptake goes down. On the other hand, if you knock down caveolin, you get less phosphocaveolin, and this will lead to increased fluid phase uptake via the CDC42-dependent pathway. And it has been reported by Rob Parton that also cavein can downregulate the CDC42-dependent pathway. So you may have co-regulation of several pathways. And as I mentioned, particles can induce their own uptake and induce intracellular changes due to their multivalent binding and cross-linking of proteins or lipids on the cell surface. For instance, we showed some years ago that uptake of ricin B, the binding moiety of the toxin ricin, coupled to quantum dots, this occurs by a macropinocytosis-like mechanism, which is quite different from what we see if you look at only uptake of the ligand. So you can expect differences. And it has been shown by Rødmer and co-workers that cross-linking of glycolipids can also lead to changes in membrane curvature. Here they used Shiga toxin, which binds to the lipid GB3 to cross-link GB3, and they saw changes then in membrane curvature, which will, of course, then induce pinching off as well. So when you cross-link glycolipids, you may do more than curving the membrane. For instance, if you add Shiga toxin, like people did in the last experiments I showed you, or antibodies to GB3, then you actually induce calcium transport across the membrane. You induce sick phosphorylation, it's a small tyrosine kinase, and this will lead to a dissociation of a complex consisting of phospholipase A2 and annexin A2. And when you get free active phospholipase A2, that can, of course, induce membrane tubulation and change transport in itself. And you can even see changes which occurs upon cross-linking of glycolipids at the cell surface. This is a control cell where you have annexin A1 in green, the lysosomal marker LAMP1 in red. And after cross-linking of the lipid GB3 at the cell surface by adding Shiga B, you can by eye see large changes due to the signaling that occurs across the membrane. And to optimize delivery, people ask which endocytic mechanism is involved in uptake of a given particle. For instance, is cholesterol involved, and what does this tell us? And then, of course, one should remember that cholesterol is not only important for caveoli, but several endocytic mechanisms are cholesterol dependent, and that cyclodextrin and genistein are not caveoli-specific inhibitors, a claim which you often see. And I'll show you a toolbar that we made some years ago to try to help out the confusion, where we, for instance, list that when you give this compound, methyl-beta-cyclodextrin, which depletes cholesterol, that can inhibit both micropinocytosis, clathrin-mediated endocytosis, as well as clathrin-independent uptake. And one should be careful, because if you extract too much, you will get leakage of potassium across the membrane, which is a much more sensitive measure than to look at leakage of proteins, which requires quite big holes. And this is to show the role of cholesterol in clathrin-dependent endocytosis. If you look at clathrin at the membrane, you can see different structures, quite flat ones, more invaginated ones, and almost pinched off structures. And in a control cell, you have a lot of the invaginated ones. But if you extract cholesterol, you will get flat clathrin-coated pits. So you inhibit the uptake of clathrin-dependent endocytosis as well. And here we show that cholesterol is essential for macropinocytosis, because both membrane ruffling and macropinocytosis of horseradish peroxidase studied by EM, it's reduced here as a control cell, small vesicles with black horseradish peroxidase. If you stimulate ruffling and macropinocytosis by adding phorbol esters, you get big vesicles containing horseradish peroxidase. If you extract a little cholesterol before you add TPA, you see the cell is calm, it can no longer ruffle, and you won't get macropinocytosis anymore. And this is due to inhibited recruitment of the small GTPase rack to the membrane. Importantly, particles can end up in a different location than free ligand. For instance, transferrin Q dots, they are endocytosed via clathrin-coated pits, but they do not recycle. We don't know why, whether it could be dependent on the size. And for instance, Shiga B, coupled to Q dots, these particles do not go to the Golgi in the same way as the toxin binding part Shiga B does. And I will show you that. And also, particles may affect other pathways in the cell than those they use. Whether this is due to trapping of membrane or signaling is not known currently. This shows that Shiga B, the binding moiety of the Shiga toxin, it goes to the Golgi, it becomes co-localized with the Golgi marker TGN in a control cell. Here's the co-localization. But if you couple it to Q dots, it will stay in vesicles. It does not enter the Golgi apparatus. And I want to, therefore, mention the importance of biodegradable particles. We have earlier shown that the effects of Q dots on intracellular transport in the cells where they enter. And there was a recent publication in JBC, published in May, showing that there is a lysosomal dysfunction caused by cellular accumulation of silica nanoparticles, which is perhaps not too surprising if you fill up the cell with something which it can't really handle. And another important point when it comes to studies of uptake into cells is that one has to make sure that a given ligand or nanoparticle is internalized or whether it's only absorbed to the surface membrane, because the outcome can be quite different. So it's important to differentiate and not only talk about cell-associated particles. And there are several possibilities. One can do EM with serial sectioning, EM with fixation in the presence of a ruthenium red, which stains membrane connected to the surface, or confocal microscopy with set stacks. And this is an example of ruthenium red, where you see staining of something which one could have thought was an endosome, but it's connected to the surface. And I won't go into this. This is, of course, confocal with set stacks, which can be useful. And, of course, as we increase the sensitivity of microscopy, so we can even see, like here illustrated, red nanoparticles in lysosomes, then, of course, one can figure out at a quite different level where do the particles we look at, where do they end up. So I want to stress that I think cell biological background is very important to draw the right conclusions, that in this field it's very important with interdisciplinary collaborations in order to get to the right conclusions. And I also want to mention that we have a national competence building project led by me, which involves 10 groups in Norway from industry as well as university and hospitals. And I want to add a picture of most members in the current group in Oslo. And I want to tell you that we have a researcher position now available for studies of particles as well. And I can see I made it in time, so thank you for your attention.
Yeah, thank you very much. You were even faster than expected. So I think there are some questions from the audience. Please come in and take a seat. There are some seats available. So where's the question over there? Wait a moment for the...
Hello, thank you for your talk. I want to ask you about the sentence that you said that nanoparticles can end up into the cells, but actually their ligands can go somewhere else. So
yeah, when they are separated from the particle.
Yes, separated.
Yeah.
Anyway. How could you distinguish that by electron microscopy, for example, to be able to tell the difference between of a nanoparticle, where does it end up, and like?
Well, I think, I mean, it's useful to use a combination of technique. What we did in those experiments, well, I showed you that Shiga B goes in a different place than Shiga B coupled to nanoparticles, but we have also shown we actually have some genetically engineered toxin molecules that can be sulfated in the Golgi and glycosylated in the ER. So if we add those in combination with nanoparticles, we can see whether you now prevent Golgi transport, because you won't get sulfation in the Golgi of your genetically engineered molecule. And that is what happens. For instance, we can see that uptake of Q dot can increase transport to the Golgi of certain molecules, whereas actually others are inhibited. And why there is a difference, we don't know. We know that the two types of molecules we looked at when it came to transport one is dependent on rafts or cholesterol, one is not. So depend on the particle you take in, it could be that it interferes with different types of transport.
Can I ask one more question? On what properties does it depend, the route of pathway, like a nanoparticle? What properties does it have in order to take a specific route? Because apparently there are so many routes, as you showed us in the image, like you showed us many different endocytic pathways, but how do we know which one a nanoparticle we choose?
Yeah.
Like, maybe it's dependent on size, but like a similarly sized nanoparticle, which route could it take?
Yeah, that's the challenge. I mean, of course, you have different ways then to study it, but you just have to make aware of that. If you knock down a given protein like caveolin, you may affect more than caveoli. You may affect CDC42-dependent uptake as well. And that's also why I added that there is good evidence now that when you add a particle which can cross-link a certain protein or a glycolipid, that may induce a different uptake mechanism than the ligand on the particle.
Okay, good. Thank you very much.
Okay, thank you very much. We thank the speaker again. And we come to the next presentation.